Good evening and welcome to tonight's event. We're so pleased that you could join us for our fourth virtual lecture highlighting our impact and expertise in the COVID-19 pandemic. I extend a warm welcome to all of our staff, students, alumni and supporters from around the world who've joined us this evening. For those who don't know me, my name is John Holloway. I'm Professor of Allergy and Respiratory Genetics, as well as Associate Dean Research for the Faculty of Medicine here at the University of Southampton. So over the next hour, we'll run as follows. I'll give you a brief overview of Southampton's response to the pandemic, and then we'll hear from our three speakers, Professor Rob Reid, Professor Saul Fowles, and Professor Lucy Yardley. All three are at the forefront of COVID research and experts in their field. And that should leave us about 25 minutes after the talks for the Q&A session, which is your chance to ask any questions to the panel. So it's certainly been a challenging 12 months. All of us have faced unprecedented disruption and felt the effects of COVID, either directly, indirectly, and in a multitude of different ways. A source of great pride for me, however, has been seeing our university community come together to tackle the pandemic. Whether it's been our staff, our students, members of our city, and alumni across the globe, we've recognised that we have an abundance of talent, ambition, and innovation here in Southampton, and our contributions have made a real difference and continue to do so. From the outset of the pandemic, universities like ours have demonstrated that scientific expertise can generate solutions to the crisis. Academic expertise is at the forefront of the coronavirus response, with students and staff providing vital support to communities and academics progressing research that is saving and changing lives. Here in Southampton, we have a unique partnership with University Hospital Southampton, and our strengths in medicine, social sciences, and engineering in particular are reflecting the truly remarkable capabilities of our university. Our three speakers tonight come from across our university, emphasising this depth of research talent and expertise we have here at Southampton. We'll hear about research on human challenge studies to understand more about the pathogenesis of SARS-CoV-2, the major research programs underway to assess efficacy and safety of COVID-19 vaccinations, and examples of public health interventions to help change behaviours to protect people from COVID-19. But first, we're going to hear from Professor Robert Reid. Rob is Director of the NIHR Southampton Biomedical Research Centre and Professor of Infectious Diseases at the Faculty of Medicine. So Rob will now talk you through his role in the Human Challenge Study, which has been researching how vaccines work in young, healthy people and how this will influence new treatments. Rob has extensive experience in the use of human challenges to investigate pathogenesis of infectious disease, and has recently published a paper on the public consultation into acceptability of controlled human challenge studies with SARS-CoV-2. The results of this, which will hopefully feed into new studies on SARS-CoV-2. So over to you, Rob.
Hello. I'm going to talk to you about something that on the face of it might at first seem counterintuitive, and this is human challenge with the virus that causes COVID-19 or SARS coronavirus 2. Now, human challenge is where you deliberately infect people with an infectious agent to discover better the pathogenesis of the infection and how best to protect against it. And in fact, Southampton has got considerable expertise in human challenge. We were the first centre in the world to conduct a human challenge with the organism that causes whooping cough, and we've learned an awful lot from this suite of experiments. Likewise, we have an interest in meningitis, and we are regularly conducting infections with a novel organism closely related to the bacterium that causes meningitis, Neisseria lactamica. And we were the first group in the world to use genetically modified bacteria to study susceptibility to meningitis. But human challenge really has tremendous potential to very rapidly assess new vaccines for an infectious disease. And that's why the UK government has brought together a number of experts in the UK. The Department of Business, Energy and Industrial Strategy, via its Vaccine Task Force, is collaborating with a number of universities that have expertise in human challenge, including ourselves, UCL, Imperial College and Oxford. And we are investigators that already collaborate in a consortium funded by the Medical Research Council called HICVAC, which uses human challenge to study vaccines. So why would you do a human challenge model with SARS coronavirus, and why would people volunteer for it? Well, human challenge really does accelerate the process of evaluation of novel vaccines. For example, JCVI would like to know whether vaccines that are currently being deployed can not just prevent disease, but also reduce infectiousness and therefore transmission between humans. Human challenge allows you to rapidly test what is the best dose, what is the best dosing schedule, spacing between doses, etc. Likewise, understanding why people are vulnerable to infection with the virus is a valuable way to discover surrogate markers of protection that allow you to rapidly test vaccines in the field as well. And finally, in the interpandemic phases, when the disease naturally occurring is relatively uncommon, then human challenge gives you a way of testing new treatments and also new diagnostics. So human experimental medicine always starts with public engagement. And in fact, you probably were aware that last summer, in fact, there were lots of calls from groups of the general public for human challenge to be done to accelerate our way out of the pandemic. And in fact, in Southampton, we responded to this via our Biomedical Research Centre by doing a public engagement exercise where we learned that the public would be more than willing to take part in human challenge. And we also learned how the public felt that a human challenge programme should be done and how best to enrol and inform volunteers willing to take part in the study. So how do you do a human challenge with SARS coronavirus, and how do you make it worthwhile? Well, the first thing to do is a dose ranging study where you take the lowest dose of virus and you find out the lowest dose that can safely and reproducibly infect a sufficient number of people to make the model useful. Then you might do a reinfection study where you find out whether people who have previously been infected are protected from reinfection, because that can tell you how to use information from people who've been infected already to learn about what protects you against infection. And finally, you can do formal vaccine challenge studies where you vaccinate groups of people and then infect them to work out how well the vaccine performs in preventing infection in a standardized way. But human challenge begins with manufacture of pharmaceutical grade batches of the virus. And in fact, the UK government via the Department of Business has already funded the manufacture of a challenge strain using good manufacturing practice of the original virus that infected the UK. But obviously since then there have been emergence of variants of concern and these will also be manufactured as time progresses and once the model has gotten underway. So when you take volunteers and obtain their informed consent, it's really important to be absolutely clear about the possible consequences of being infected, because a volunteer needs to know exactly what could happen to them if they're to take part in such a study. And now we know from a lot of very detailed clinical epidemiological research done in the UK exactly what happens when people are infected with coronavirus and the consequences of infection. We now know that after an incubation period of approximately 5 days, people will become infected actively with the virus and will either experience no symptoms or very commonly very minor symptoms, initially consisting of fever, tiredness, muscle aches and cough. Unfortunately, in some people, the cough worsens and their respiratory pathology worsens, and they can get quite severe respiratory symptoms that in some people can result in hospitalization and the requirement for management on a respiratory unit. Generally, this reaches a zenith between 12 and 21 days when, in most people, the infection then gradually improves. However, some people can go on to a rather protracted convalescence, and other people can get rather difficult complications, including heart, lung, blood, and neurological disorders. Now, we know the rates at which these features of the disease are likely to occur in specified age bands. And now we know that people aged between 18 and 30 are extremely unlikely to suffer pathology after infection with the virus. And we know approximately at what rate they are likely to suffer disease. And this means that we can give volunteers for the study fairly accurate risk analysis of the consequences of their involvement in the experiment. So what would a human challenge look like? Well, it would look like this. You would screen individuals between the ages of 18 and 30 to exclude people who have any underlying infections. You would then bring them into an inpatient unit, infect them with live virus, and follow them as an inpatient for 14 days. And during that time, you would take multiple samples from the nose and throat and from the blood to study the infection in the respiratory tract and the immunological response in the blood. And at the end of 14 days, you would discharge them to home isolation and then continue to follow up immunologically and virologically. And in a putative study, the primary outcome measure could be throat swab PCR positivity on 2 separate days, and secondary outcome measures would include immunological responses and physiological parameters. But when you're designing a human challenge study, you have to have a fairly intricate plan of establishing the lowest dose needed for a successful challenge of volunteers. And there is an elaborate plan that is currently in front of the Ethics Committee for a dose-ranging study where you give the lowest dose in a small group of people, and then if there's little infection observed, you would then build up the dose tenfold, and likewise, if there was established infection of a group, you would then increase the numbers of people exposed to the standard inoculum that you've identified. And the overall pattern looks a little bit like this, and you come out of this usually by about 30 volunteers with the standard inoculum that you need to infect reliably and reproducibly and safely a sufficiently large cohort of people with the virus. And then once you've done that, you can then move on to a vaccine challenge program where you would either take a vaccine or no vaccine, a placebo, and then 28 days later infect people with the standard inoculum that you've identified to be safe and reproducible. And you would measure by quantitative means the amount of virus within the nose and throat after an inoculation. And you would then be able to compare either vaccine and placebo or novel vaccine and putative vaccine to look at the relative efficiency of inhibition of the viral replication in the respiratory tract. So I'm going to stop there. There are numerous people who've been involved in getting our program to this stage of presentation to the Ethics Committee, and they're all shown here. And I'd like to thank all of them. I'd like to thank you and welcome any questions. Thank you very much.
So thank you, Rob. It's a fascinating insight into what a human challenge study would look like. And I look forward to those developing over the next few weeks and months. So our second speaker this evening is Professor Saul Faust, who is Professor of Pediatric Immunology and Infectious Diseases in the Faculty of Medicine at the university. Professor Faust's expertise is around the work to bridge clinical and laboratory interface, and he heads our clinical research facility, where a lot of our experimental medicine research is undertaken at the university. So Saul has been heavily involved in a number of national clinical trials for vaccinations on COVID-19, including the widely publicised Oxford vaccine, as well as the Janssen trial. So many types of vaccines are likely to be needed to end the pandemic. And Saul will give us an insight into why we need so many vaccines and how we know they are safe. So Saul, over to you.
Thanks, John. I'm Saul Faust, and I'm going to talk to you today about the COVID-19 vaccine program. You'll all be very used to seeing the coronavirus on television, and the coronavirus has these spikes on it, which is the spike protein. It gets into the body, and then the immune system has to respond to the virus. And it does that by using both T cells down the bottom here and B cells to do two things. It produces anti-coronavirus antibody, and the T and B cells provide long-lived memory. That means next time we meet coronavirus, we can remember it and make more antibody. Now, when we have a vaccine, what we're trying to do is simulate that so the body can remember coronavirus without actually having to be infected. So what do we need to know about any new vaccine? We need to know they're safe. We need to know that they can make an immune response to what we want. We need to know how long protection lasts, whether the vaccine actually does provide protection against infection, and whether there's a risk of enhanced disease. Are boosters needed, or are different technologies best combined in sequence? We call that heterologous prime boost, using different vaccines, one after the other. Now, there are many different types of vaccine technology, and the UK Vaccine Task Force has invested in 4 different types, or buckets as they call them. The first is the replication incompetent vector vaccines. This is the AstraZeneca vaccine and the Janssen vaccine. And what these are cold viruses. So the Janssen is a human cold virus, Oxford is a chimpanzee cold virus, and the cold virus has been inactivated, so it can't cause a cold in the body, but what it does is it expresses the spike protein on the outside when it gets into the body, so that the immune response is a combination against the cold virus, but also against the spike protein as well. The next group of vaccines are the RNA vaccines. They haven't been used against infectious diseases before, but they have been researched a lot for anti-cancer vaccines. And what Moderna and Pfizer have done is put a piece of the RNA message inside fatty or lipid nanoparticles, and this is injected into human muscle, and the spike gene message then is translated into protein in the body, and the body makes an immune response against the protein, but aided by the lipid particle that helps stimulate the immune response. So that can't be incorporated into your DNA. It's only message that gets turned into protein, and the messages just disappear, like Snapchat messages or Post-it notes that you chuck in the bin. A more traditional vaccine technology are the Novavax and Medicago GSK vaccines, and these are recombinant, so made in the lab, spike protein stuck to an adjuvant or particles that the body then is able to make an immune response against. So the combination of the spike protein and the adjuvant stimulate the immune response to produce that antibody and then memory. And the final type of vaccine that we're using in the UK are the inactivated viral vaccines. So this is a very old-fashioned technology where the coronavirus is grown in the lab and then it's killed and inactivated, and the whole cell killed vaccine is used to stimulate the immune system as the vaccine. And that might be good. We don't know this for certain, but what it might do is it might avoid the problem of spike protein mutations that's been in the news recently. So the UK Vaccine Task Force has, by the UK delivering the vaccine research programme, also signed contracts upfront for nearly 300 million, if not over 300 million doses of these different vaccines. The UK government having taken the financial risk of delivering the trials, and then if the trials are successful, we then have the vaccine to deploy in the UK population. So this is what it looked like when we started off, and we started off by doing the Oxford vaccine trial in Southampton, and the university very kindly lent us Jubilee Hall on campus, and you can see that in the top picture of the circuit, people came in, and they were able to be socially distanced as they met the doctors and took part in their trial, and some pictures of the team in the bottom plates. Now, after the Oxford trial, we very rapidly realized that to deliver the Vaccine Task Force program, we were going to need to take a sort of unified national approach, and all 18 regions, 15 in Britain and Scotland, Wales and Northern Ireland, have taken part in a national network for COVID vaccine delivery. And there's a national registry system for signing up to take part in the vaccines, and the capacity for that large-scale recruitment involves research coordination and governance, training of staff, finding location, logistics, providing 24-hour on-call for the participants, and being able to actively monitor subjects for infection, both when they have symptoms, because it's when they get symptoms that we find out whether the vaccines work or not. Of course, the Oxford study, as you might have seen in the news, has also been swabbing people every week to find out whether the vaccines prevent transmission. So within Wessex, we took a system-wide approach to this with the National Institute of Health Research and NHS. We were supported by the Hampshire and Isle of Wight and Dorset Integrated Care System. This is the local NHS management who provided logistics, project management, and we linked in with primary care, who have been brilliant at supporting the trials. The vaccine hubs are run by GPs and hospital doctors and people, research nurses from all over the system, and all of our participating trusts in Wessex. All of the research goes through the lead contracting trust in Southampton, which makes it much more streamlined and actually a brilliant model for the future when we want to do trials in more than one place. And it's managed by a joint pan-regional governance board that includes the NHS and research systems. So we're in a position now where we have 5 vaccines that are either deployed or pending deployment. And it's really important that people understand the data. The first thing is that all the vaccines prevent hospitalization and death. And so far, of the vaccines that have been tested, only the AstraZeneca Oxford vaccine is the one to demonstrate some definite effect on asymptomatic carriage, i.e. transmission. And that's because it was the only trial to swab participants every week and actually find out whether people without symptoms who've had the vaccine still carried coronavirus. But we can't compare the trial data directly. You'll see lots of different amounts, 62%, 95%, these figures being bandied about in the news. None of these trials are directly comparable. The first thing to say is that the mRNA vaccines, the Pfizer and Moderna vaccine data, was the maximum immunity level after 2 doses, 3 weeks apart, taken with a very strict definition of COVID. There was no basis, incidentally, for that 3 weeks, other than it being faster to give and for them to be first to market. The AstraZeneca, Oxford and Janssen trials showing lower headline immunity, this is partly because cases were included with mild symptoms. You only had to get a cold to come in for a swab on those studies. And so you can't compare those studies directly with the mRNA vaccines. And we also know that the adenoviral vector vaccine, so Janssen and AstraZeneca Oxford, are known to build immunity with longer interval between doses. And that data is now in the public domain with papers published specifically for the AstraZeneca Oxford vaccine that show that the longer dose interval is beneficial for immunity. So deployment in the UK is on the direction of the Joint Committee of Vaccination and Immunisation, which is not the government. This is an independent scientific committee, and they have prioritised the groups and established the dose schedule. They've prioritised the groups being immunised because these are the groups that whereby 99% of people who are at risk of death will get their vaccine in the first round of the immunisation programme. Those are people over the age of 50 and people with other conditions and healthcare workers. And the dose interval to 12 weeks was a change to the single dose schedule was because of the modelling data based on both the AstraZeneca Oxford studies, but also the data available from Pfizer and Moderna. It's caused unnecessary controversy in the press. It's definitely the case that this is the way that we will reduce the pressure on the NHS by taking out the people who are going to be hospitalised or have the risk of death. And the trials in Southampton contribute to these policies. We recruited all 240 more than 70-year-olds in the phase 2 Oxford trial, And we've, of course, recruited to the main phase 3 trial where the different length between doses was also studied. So there's an ongoing need for clinical trials of different vaccines and technologies to ensure the global supply, both for the optimization of protection using different vaccine combinations or in different age groups, different clinical conditions, but also to address any issues caused by virus mutations, to make sure that we have a global manufacturing supply chain that works for the globe, not just Western countries. And we really must avoid vaccine nationalism at all costs if we can. Finally, just a couple of words about public confidence. Clearly, the regulatory process needs to be seen to be done, and this has been undermined internationally by politicians and some scientists. People in the UK need to know that the UK Medicines and Healthcare Regulatory Agency, or MHRA, are global experts in medicines regulation. We used to do all this work prior to Brexit for the European Union. It's a huge disappointment to us scientists working in the field that politicians and other scientists are making statements in the press and internationally without understanding the data or the process. From a clinical perspective, most of the public are likely to accept the pandemic vaccines to protect themselves and others, as people do every year for flu. And in fact, in the UK, the children's flu programme is to protect the vulnerable in society, not the children themselves. And this has been hugely successful. We are already seeing that the programme proceeding successfully has led people to be confident that they're doing the right thing in having a vaccine. Hesitancy is a problem. The media can help by not focusing on it, as oxygen definitely makes low-level anxiety and probably overall hesitancy levels worse. We are a bit worried about the hesitancy in the Black and Asian minority community in the UK and US that's clearly driven by social media and misinformation. None of the myths are true that are circulating. We need clear, transparent communication from the JCVI, NHS and government. And ultimately, governments globally will have a choice and do have a choice as to whether to regulate social media misinformation and not just label it. They could choose to do that. Other interventions, probably much more controversial, much more complicated to say, well, people who've had vaccine can do this, but you can't if you haven't had a vaccine. And that could be perceived as compulsion, which is likely to be detrimental overall to public opinion in the UK. Healthcare worker immunisation might be one that we need to treat as a special case. But again, that would be very difficult to do and would need considerable consultation and workforce support. So finally, which vaccine should you have? Well, I hope I've reassured you, any deployed vaccine that has or been or will have been fully evaluated by the MHRA will be one that you can take confidently. The main uncertainties are not about safety. They're about how long immunity is going to last or how often we need to give them, how well they're going to work in different age groups or the immunosuppressed and so on. The current UK dose schedule is evidence-based and aimed at reducing deaths and admission to hospital at this moment. Please take the first vaccine offered to you. I certainly did. Thank you very much for your time.
Thank you, Saul. That was a whistle-top tour of COVID vaccines, and I know we've got a lot of questions coming in on vaccines for after our final talk tonight. So our final speaker is Professor Lucy Yardley, OBE, and she was awarded her OBE in the Queen's Birthday Honours last year in 2020 for her services to the COVID-19 response. So Lucy is Professor of Health Psychology at both the University of Southampton and at Bristol, as well as sitting on the government's Scientific Advisory Group for Emergencies, or SAGE. So Lucy has provided evidence-based advice and expertise throughout the pandemic, particularly in relation to supporting people to implement measures designed to reduce the impact of the virus. So Professor Yardley will also talk about her research on preventing infection transmission using germ defense and how we keep safe by maintaining preventative behaviours during the vaccine rollout. So Lucy, over to you.
Hello, I'm Lucy and I'm going to be talking about how I was providing behavioural advice to SAGE this year. And I was providing the advice in my capacity of co-chair of what's known as SPI-B, which is a subgroup of SAGE. What is SPI-B? Ironically, it's actually the Scientific Pandemic Influenza Behaviour Subcommittee. So even though COVID, we now know, is not influenza, SPI-B is a historical name from it providing advice in the last pandemic. And what SPI-B does is produce papers giving rapid advice and The papers are discussed at SAGE, and once they've been approved, they're circulated throughout government or to all the relevant departments. And then within a week or two, they're published so that the general public can see what we've said as well. Now, we provide advice to SAGE, but also to other areas of government and to subgroups and committees. So, for example, SPI-M, which is the modelling subgroup, EMG, the environmental subgroup, JCVI, advising on vaccines, and direct to government departments. So I've learned lots of initials of government departments that I never used to know. So Cabinet Office, Department of Health, Ministry for Housing and Local Government, and so on. How does SPI-B work? Well, we're a group of experts from lots of different behavioural disciplines, and we meet online, of course, and we discuss what advice we can give based on behavioural theory and evidence. But that's where the problems start, because when you're dealing with a completely new pandemic, there is very little evidence that you can use. So you might go to the past research, but you're worried about whether that's actually relevant to the current situation. You can look at what's happening in other countries. You often only are learning about that through, the media, and you don't know whether the cultural context is so different that it's, again, not relevant. You can carry out rapid surveys. That relies on people answering the survey, which not everybody from every sector of the community wants to do. And it relies on people being honest, which, again, you can't always rely on. And then you also rely on rapid research. And that's where my own germ defence line of research came in. So what's germ defence? The germ defence team developed the first online intervention worldwide that was able to show that you can use hand washing to reduce spread of infection in the home. So when we did this study, although it was advised to wash your hands and it was hoped that that would actually reduce infection, we didn't know whether that would actually reduce spread in the home, for example, where everybody's living together. But we trialled our digital intervention to get people to wash their hands in 20,000 adults in the last pandemic and in seasonal flu years after that. And it did work. It reduced infections in the user and family members, both the incidence, frequency, severity, consultations and antibiotic use. So that was a more objective measure. And that led to me being on SPI-B at the last pandemic. So this is germ defence, and you can see how we tried to encourage people with the feeling of disgust, because emotion such as disgust is very powerful for changing behaviour. So we tried to make people feel that an unwashed hand was disgusting. So on to the COVID. In early 2020, all the former SPI-B members were reassembled, and I became co-chair in March. At the same time, we got rapid funding from UKRI to adapt and disseminate germ defence for the new pandemic. So we updated it for more behaviours, not just hand washing, but things like social distancing, mask wearing where appropriate, and so on. And then we launched it to the media. And we launched a whole raft of research at the same time. So we published a rapid paper in the BMJ that was a review highlighting that transmission was likely to occur in the home, and that we need to do things about it, and that something like germ defence could help. We collected user feedback. So we rapidly got 100,000 users, and thousands of them volunteered to give us feedback on how to improve germ defence, which we used. So we were constantly updating it as we learned more about the pandemic and about what people needed to know. We also did a quick study to look at what about if people absolutely couldn't socially isolate in the home, what about if you offered them accommodation outside the home, which is particularly relevant for people with lower levels of income. And we did a systematic review proving that germ defence is still the only digital intervention worldwide with evidence of effectiveness. And a result of this raft of research that we did was that Germ Defence gave valuable input to 3 of the SAGE papers that I contributed to, including when I took a step back and said I had conflict of interest and other people put it in. And it was the only evidence on what people were currently doing in the home, because it actually asks people what you're currently doing as well as what you intend to do based on the Germ Defence advice. And it had also, we had collected all the user feedback about what were the barriers to doing more and how we could overcome them. And the other study was also the only evidence on what the acceptability and barriers were to self-isolation outside the home. And with the help of all our users, volunteers translated it into 20 languages. We've disseminated it worldwide. We've had over 500,000 logins. And it's being rolled out in a very rapid trial that was prioritised by Chris Whitty as an NIH urgent public health study. So all of the practices in England have been randomised to either disseminate it to their patients immediately, or it was actually either before or after Christmas. And we're going to look at the impact on infections using routine data. We've also got a trial in South America. So in conclusion, lessons for the future pandemics? Well, I pointed out how difficult it was to have evidence that we sorely needed right away at the beginning of the pandemic. And if we had more, for example, digital observational data collection going on all the time, we wouldn't be relying on responses to surveys. And I would also like to see permanent panels of public and patient involvement engagement groups from all sectors of the community, so that in future we can involve them all the way through in giving us really rapid input, feedback and co-design.
Thank you, Lucy. So really good to hear something about the non-vaccine or non-infection part of the response to the pandemic, because all parts of scientific expertise have been important in creating an effective national response to infection. So I'd just like to thank again our 3 speakers, Rob, Saul and Lucy. And we're now going to have our question and answer session. So as a reminder, if you would like to ask a question, then just type your name and question in the Q&A box, which you should see on your screen. And maybe just as a starter, Saul, perhaps you would like to comment on how has the pandemic response brought the university together with our regional community?
Yeah, John, I think in a huge number of ways, as you know, and on a different topic, the Southampton testing programme has engaged the city and NHS and university. But for the vaccine studies, the university were absolutely fantastic. When we first needed a venue for the trial in the Jubilee Hall, and then ongoing when we were needing to move out for teaching to go back into the hall, finding a space at Wide Lane to carry on the programme before we found a home in the NHS in the city centre of Southampton. An unbelievable effort by everybody across both organisations to help us get the trials done.
Thanks, Saul. So we've received a number of questions, as you can imagine, about the efficacy of vaccine trials and the delivery of different vaccines. So Julianne Bauer would like to know if the vaccines are safe for those who have anaphylactic reactions to food or to other allergens. And are there any contraindications for vaccines, either people with allergies or people taking other medications?
No, I mean, it's hard to talk about individual cases, but in general, both of the deployed vaccines in the UK are now thought to be very safe. There was a concern about very anaphylactic people at the beginning who carried an EpiPen, but even that's been relaxed now, and people should be confident about going to a vaccine centre and telling the doctors involved what their allergies are, and they'll be given the right advice and appropriately immunised.
Okay, so Rob, I mean, you serve on JCVI, so providing advice to government about vaccine policy. Rebecca Robbins is interested to know what she can tell her colleagues who insist the vaccine is, compared to other inoculations, relatively untested and presenting a high risk. What can you say about the process to reassure people who are perhaps worried about that?
Okay, so on JCVI, I'm privileged to get access to a lot of information, and the British public recently was told by MHRA, our medicines regulator, that the signal so far for safety for these vaccines, based on millions of doses given, is exceptionally good. So, that's extremely good data. Usually with a vaccine that's considered relatively unsafe, we would expect a safety signal in the order of 1 in 100,000. And yet we've had millions of doses administered, and the safety profile so far just looks like local reactogenicity at the injection site, and some flu-like symptoms afterwards and muscle aches, and that's it. We haven't had any of the difficult neurological side effects that we were, that were signaled very early on in the vaccine studies.
Saul, is there anything you'd like to add on that?
No, I think there was a question about, was the program too quick? No steps were unturned. So all of the steps that would normally go into drug development were carried out at pace and scale, and sometimes in parallel, but nothing was left undone. So the phase 1 trials were big, the phase 2 and phase 3 trials were enormous. 44,000 people in the Pfizer study, the Janssen studies. These are much, much larger than normal, even before we deploy. And I think the point that Rob's just made is that, even if people were worried that things had been too quick at the point where the MHRA gave regulation, and none of us were worried, now there are millions of doses that have been studied in the population. And that's Rob's point, that the MHRA have reassured us last week that we now know a lot about these vaccines and they're very safe.
So if the vaccines are safe, of course, we're getting quite a few questions about their effectiveness against variants of the virus. Is it the Kent virus or the South African virus or the Brazilian virus? Guy Blythman, for example, is wanting to know whether these different variants could be resistant to current vaccines, and how many variants are we likely to see over the short or medium or long term?
So if I can take that one initially, coronavirus, like other respiratory viruses, is capable of profound and continuing variation. And it's important to understand that although a virus can become selected so that it's relatively resistant, if you like, to the antibodies that are made by the host, it's extremely unlikely that it would be absolutely resistant to the immune response. And so what we're seeing is that there are some individuals who have relative susceptibility to mild forms of the infection, but do not go on to get severe disease. And this makes complete biological sense, that the virus will have selected for transmission between humans, but the immune response that, remember the immune response has got many different components. Some of them will prevent mucosal replication in the surface of the nose and throat, and others will be to do with the systemic spread within the body of the virus. And it appears that some of these components are, the virus can become relatively resistant to, but not all of them. So it's relatively, it's very likely that we will continue to be protected even in the face of variants which may emerge. You'll be aware that already we're planning nationally in our program to work with companies to introduce minor changes in the existing vaccines so that they would cover all of the potential variants that we are likely to see as they emerge.
And the only thing to add really, John, is that to follow on from Rob, all of the vaccines stop hospitalization and death. But what we're very unlikely to be able to do is make coronavirus go away. This is going to become a normal respiratory virus that changes and develops. But once we've got a baseline level of protection in the whole population, which we'll have by the mid to the end of the summer with the first round of immunization, we should be under much less pressure. Some people might end up getting more severe disease, just like flu. But actually, the vaccines we've got look pretty good at stopping people coming into hospital.
So if we're going to be living with coronavirus for some time, Lucy, how do you think we can sort of embed the behavioural change that we've encouraged people to adopt in the midst of a pandemic for the long term?
That's a very interesting question, because it's linked to the cultures of different countries. And what we've seen is that some countries' cultures are easier to use to reduce infection spread. And we really need to change some of our habits. Apparently, one of the bad habits that people have in the UK is going to work when they're not feeling well. And I'm sure that's a habit that we'd all love to change in both ourselves and our colleagues. So yeah, there's all sorts of changes like that. I think we all are habitual hand washers by now, that are hopefully opening windows, which we now know is really important. I think it's also a question of sort of making sure that if you are symptomatic, staying at home and not infecting anyone, not just people at work, but anyone else.
Thanks for that, Lucy. So we've had quite a few questions about reinfection, if you like. So Laurie Marsh and Olivia Aspinall and others have asked questions about, if someone has antibodies, whether because they've had a prior infection or because they've been vaccinated, what do we know about their chance of being reinfected?
So I edit the Journal of Infection. I'm the editor-in-chief of the Journal of Infection, and actually this is the topic that has attracted the most number of original publications coming into the journal over the last few weeks. And actually, you can get, it seems, a second infection, but it's actually quite unusual. You have to remember that we've had now, a very large number of infections worldwide. And actually, the reporting rate of severe second infections is very, very low indeed. However, that's not to say that it can't occur. And it does appear that people's immune system its immune response to the virus can wane a little bit. But these second infections, if they are real, tend to be very, very much milder than the first ones. And that's a sort of consistent report that we're starting to see. So yes, you probably can have a second infection, but it's likely to be milder.
And again, in terms of prioritization of groups for vaccination, there's evidence that perhaps men are more susceptible to severe disease than women. Why wasn't there a sort of gender prioritization suggested for vaccine rollout?
You mean by the JCVI?
Yeah.
Okay, so you're right. There is a small signal for increased disease rates and severity of disease in males, but remember that there are multiple factors contributing to whether someone has severe disease or not, and it's rather difficult to unpick the role of those so-called comorbidities in this question of why men get slightly higher severity rates than women. And JCVI was not confident that it was just your sex that was responsible for this difference. And in any case, JCVI doesn't really like non-equity in how it distributes vaccines. So those two things, it was decided that we would not separate out men and women in the vaccine program. Clearly, we did set out a rank order of prioritization, but it was basically age stratified, and it included extremely vulnerable people with certain conditions, as the public will be aware of now.
Okay, thank you, Rob. So we've got a few questions about long COVID, or disease symptoms that persist for a long time after the infection is resolved. One of you wants to comment on whether into the modelling of the benefits of vaccination, we're taking into account prevention of long COVID as a benefit.
I mean, I don't mind starting off with that one, John. I mean, 13 weeks ago, literally 13 weeks ago to yesterday, we didn't have any vaccines, and we didn't know whether any of the vaccines would work at all, let alone have such high rates of infection, or that we'd be able to be sitting here saying the NHS has immunized nearly 13 million people in the population. So I think these are very interesting discussions, but actually the policy the JCVI had to pursue was, how do we take the pressure off the NHS as fast as possible, and how do we roll out vaccines equitably on the basis of stopping people getting very severely ill. I think the questions about how we use vaccines in different populations and to prevent longer forms of the disease are for the future, really. Once we've given everybody their first round of 2 doses, who wants it in the adult population, we can then work out how to immunize children, if they need immunization at all, once we've got the data from children's studies, how to immunize people with immune conditions, and whether the different vaccines will be better in different populations, and whether the vaccines have an impact on long COVID, as you say. But I think this is about priorities in the situation we're in at the moment. And those questions are for the future, really, not for now.
Okay, thank you, Saul. So, Lucy, a question from you, from Christopher Kanjanuri. So he's saying that since people take notice of large numbers, do you think that we could express the R number in a different way? Should we express it as a multiple, so it's R100 or R1000, rather than a number like 1, which people think, oh, 1's not that bad?
That's an interesting approach. I wish it was that easy. Actually, people don't really respond in behavioural terms very much to numbers at all. What they tend to respond to is what they can see close to home. So what they see as is happening to their relatives, their friends, how they're behaving. So all the way through the summer, last summer, where, on SAGE, we knew that it was coming back again in the autumn, and that we needed to try to get people to understand how to reduce the risk and keep it under control. The problem was that I was really sure that whatever we said about the science behind it, it wasn't until people actually saw the hospitals filling up again that they would really take notice and take it seriously as a risk to themselves and their families. And unfortunately, that's exactly what it took.
If I can add something, John, I mean, if it might help people to know what the R number for other infectious diseases is. So, for example, measles would have an R of 15 to 20. Whooping cough is about 17. So actually, although we talk in terms of an R of 3 for coronavirus being frighteningly high, compared to other infectious diseases, it's not. Anyway.
And how have we dealt with those infectious diseases with such a high R number?
Well, we've used vaccines. I mean,
Exactly.
That's the point about high R numbers, is that you have, the higher the R number, the more important it is to develop a vaccine as quickly as you can. So fortunately, measles vaccine and whooping cough vaccines work. We've managed to eradicate those diseases. Oh, well, reduce them at least.
Thank you. Saul, we're getting a few questions on mixing of vaccines, if you like. That should we have 2 doses of the same vaccine, or does it matter which which vaccine we get for our second dose. Richard Baldock is asking, do you think that deliberately mixing vaccines might be more effective as a way of generating a strong immune response?
So we've known for a long time that giving different classes of vaccines as a sequence might be better by using one vaccine to prime the immune response and then another slightly different mechanism to give the boost. When you're developing new vaccines, companies can only test their own product. So they have to test, they have to decide on the dose and the interval between doses, and they make those decisions, and that's what they're stuck with. But now we're in the next phase with publicly approved vaccines. Government can look to actually asking the question, what's scientifically the best thing to do? So as you know, we're testing the current Pfizer and AZ vaccines one after the other in both combinations against the normal regimes, but also we'll be hopefully testing the other vaccines as they come online, so Novavax, maybe Janssen, and others as the MHRA approve them and as Moderna gets supplied to the UK in exactly the same way, because actually we need to understand how best to deploy the vaccines in the real world. And we know, but as of now, short answer, people will get given the same vaccine that they've had on the first program unless they're in a trial, unless there's some catastrophic logistic mix-up that means something else has to happen. But we think it will be safe, even if logistics causes some people to receive one after the other. Our intention nationally is to study it before that happens.
Okay, thank you. So there's a few questions about the difference in immune response from vaccination compared to natural immunity after infection. Sarah Fair is asking about how protection offered by vaccines differs to naturally acquired immunity. And Bo Priestley is asking whether there's a role for natural immunity caused by infection to play in controlling the pandemic. So in all those people in the population that have already had an infection, will they contribute in addition to those that are being immunized in terms of generating our herd immunity?
What an excellent question. And of course, natural immunity is such an interesting phenomenon. And it is certainly true that infection with coronavirus, as we were just discussing, at least attenuates subsequent infection. And it's going to actually be very interesting with this particular viral infection to observe the relationship between the herd immunity conferred by vaccine use and the naturally occurring immunity that's caused by natural exposure to the disease. How do they differ? Well, in vaccination, you generally will use a limited number of what are called epitopes, in other words, parts of the infecting agent, to induce a potent immune response. In natural infection, you develop a more global, if you like, response directed against many different components of the infecting agent. In fact, vaccine-induced immunity tends to be pretty potent. Naturally induced immunity tends to be more global and, if you like, a holistic immune response. But the two are cognate actually, and natural immunity across a population does enable you to get faster to a herd immune effect. I'll stop there.
Okay. So, Lucy, we know that misinformation is prevalent with modern media and social media, and Don Rolt would like to ask how the scientific community can stop the misinformation pandemic as opposed to the viral pandemic, especially when it can be spread by theoretically people in positions of trust.
Yes, that is a very important and difficult question. Scientists alone can't stop it, and it's going to have to be government and citizens in general that stop it. Scientists can play their part, and it's really important for scientists to sort of be heard. And actually, the media has been really good in the pandemic in terms of giving scientists a voice. So that certainly helps. But if people are in a bubble on social media, they may not hear that voice. And so in terms of sort of stopping these bubbles of misinformation, which are really they're really impervious to sort of scientific argument, I think that's not something that scientists can do. I think that's something that the whole society is going to have to tackle.
Okay, thank you. So we're coming to the end of our time. So perhaps as a last question, Mark Spearing is asking that as we approach the anniversary of the first lockdown a year ago, what does the panel think that the new normal will look like for coping with COVID a year from now? You know, how has society changed permanently, and what will we be doing differently in perpetuity?
Who do you want to go first, John?
Which one of us do you want to answer that?
Saul, you start.
All right, so I'm going to take an optimistic view, John. I think that we will, if we take foreign travel out of it, because who knows what the globe is going to let us do, or the government will let us to go in and out. But I think if once we've immunized our population, we will have a situation where coronavirus becomes like a normal respiratory virus. We will have some people who get sick, who maybe have had vaccine or maybe haven't, but we'll be able to handle it in the healthcare service, and we'll be able to control the infection with vaccines like we do with flu. And I'm hopeful that we'll have a sort of roaring 20s response after the 1918 pandemic, where things will be able to go back to pretty much normal life, albeit we're going to take some time to get there because of anxieties and concerns and social media and the internet that might hinder us getting to there quickly. But I'm going to go for optimism and back to normal within a few years.
Good. We like to hear optimism. Lucy, what do you think?
I'm also going to go for optimism, but I'm going to go for better than normal, because I think behaviourally, in the past, we've had an attitude to infection that is similar to what we used to have to what are called accidents. So if you go back a century ago, accidents were a really common way of being killed or injured, whether it was car accidents or accidents at work, accidents of all kinds. And gradually, over the past century, we've done more and more things to prevent them happening, because we've realised they are partly avoidable, and that you can do things to reduce the risk. I think we've learned from this pandemic that infection is not just a matter of chance, it's a matter of behaviour. And there's all sorts of things that we can do in the future with testing, with not going to work when you're ill, and so on, to try to reduce the levels of infection. So I'm hopeful that in future, infections will be a bit more like accidents. We'll do more to prevent them in the future.
Rob, anything to add?
Yeah, I do. I think a couple of things are going to happen. I'm not going with Saul's roaring 20s thing. I hope that doesn't happen. I think that certainly for the next few years, there will be annual vaccination of vulnerable groups. There'll be background coronavirus disease, which may ebb and flow. But I think probably the most important thing for the future is that the government will set up a pandemic response system that does take notice of warnings and acts extremely quickly, as certain other governments around the world, particularly in the Far East, have been doing, to mitigate against pandemic spread of viruses, because boy, does it happen quickly, as we've discovered, and I think the big difference will be that we will have a much better response in the future to an emerging respiratory virus.
Okay. Well, thank you all for delivering such informative updates and answering all of the questions that we've had. It's been fascinating to hear from you all, and I'd also like to thank all of you in the audience for joining us tonight and your interest and support of the University of Southampton. So we've all been through and continue to be in extremely tough times, but hopefully we can take out some hope and reassurance from the fantastic science that we've heard about today, and the evidence of the sort of teamwork and collaboration between government, scientists, academics, and society that's leading us out of this pandemic. So if you've enjoyed tonight's talk, please do join our lectures mailing list so that we can keep you updated on future events. If you visit www.southampton.ac.uk/events and select Distinguished Lectures, you can see the list of forthcoming lectures. And finally, as you go, we would like to share with you a short video that gives a brief overview of some of the other projects from a remarkable year here in Southampton. Have a good evening, everybody, and thank you for listening.
Together, we have risen to the challenges of COVID with determination, innovation, and great care for each other, and the important work that we do in our university. I would like to thank all of our staff, students, alumni, partners, and supporters, who are all part of our remarkable community. And now, here is a small snapshot of the positive impact you have had in this very, very different year. Thank you.